yes, fish is kind of going to be the denominator, or is the denominator of my program, and the main reason is that if you study biology for the sake of biology, you learn it's too complicated and it's too full of defeats. So if you don't have some kind of passion to pull you through, and my passion is fish, pick your own. Please. Okay, let's go on a quick time trial if I can. Get some ink. Can you see the red one up there? Okay, so Big Bang. 4.5 million years ago. Let's go and make a timeline all through the history of our planet until today, this very moment. We have approximately 4.5 billion years on this scale. Thanks to one of our colleagues at our department, Minik Rosing, and his colleagues actually, we go up here till approximately 3.7 billion years ago. This is the first evidence of life. Most likely occurred a little bit sooner. But this life was just a very simple unicellular organism, right? A microorganisms. Then we actually need to fast forward up until, give or take, 0.5 billion years ago, before we have the first real evidence of truly multicellular organisms. So this is the first time two cells merge and just start to divide labor in a very simplistic way that is only slightly more advanced than one single cell. And then within this narrow window is when everything else that have ever existed evolved. So us, all the multicellular organisms, plants, trees, animals, they have evolved in this narrow window. So it's just to illustrate that trying to understand life of eukaryotic or multicellular organisms without doing it in the light of the microbes that were here for so long and shaped our planet and our ecosystem and our living conditions makes no sense. And hopefully the rest of the evening will add to that. So for people who don't like analog approaches, here's just, I didn't know if I had a whiteboard, but it's just to sum it up with the units and the scale, right? These critters have been here all along, and we only just arrived. So show some humility to the microbes. So what is hologenomics? Veronica told you what the holobiont is. So hologenomics is the research field that we are using when we study holobionts. And let me just give you one of our pet or favorite examples, which is the vulture. So the background of most of the researchers at our center is evolution. So we're evolutionary biologists. We're actually not microbiologists at all. We didn't really think about them for the last few decades, but now we've come to like them. So think about the vulture. We know a vulture has evolved to tolerate extremely toxic meat in these carcasses. And actually, it's a very extreme level if you compare it to any other vertebrate species. I mean, there are many other scavengers that eat dead things, but not to the same level of the vulture. And what do you do if you're an evolutionary biologist? 10, 15 years ago, you sequence the vulture genome, and you think, oh, all the answers must be in the genome of the vulture. There must be some genes that have mutated and evolved to tolerate these toxins. And people did that, and they could not explain the whole thing about why this vulture is able to tolerate these toxins. So it wasn't really until a few years ago when they said, oh, maybe we should also look at the microbes. So by looking at the microorganisms that live actually, the intestinal tract of a vulture starts on the face, on the outside of the face, because it stick it up the carcass when it's a little less degraded than this one. Then they could actually see, by looking at these microbes, first the vulture has evolved its own genome in a way where it tolerates microbes that would probably kill all of us in this room if we tried to eat this carcass. And secondary, they also found microbes that are probably able to detoxify some of these toxins, making them tolerant to eat this resource. And then why is that interesting? Because isn't that just a microbe that came in and then it was helping the vulture? But we don't see that in any other vertebrate species at this level. So to us, it suggests that it's evolution in place, that it's some kind of time, an ancestral species to modern-day vultures evolved its own genome, giving it the ability to collaborate and recruit microbes that help them gain this new trait of being tolerant to toxic meat, and then it can explore this niche that no other vertebrate can. So one more example here. So this is just one of many species that we would consider being poisonous or toxic. So in this case, it's a newt, the red-bellied newt, which is poisonous to avoid predation, basically, right? So that's a very logical evolutionary adaptation, that if you don't get eaten before you reproduce, you might have a chance to survive and evolve as a species. However, when you look closer to what's going on here, the newt is actually not poisonous. If you look closer, just like Veronica said, if you look under a microscope on your piece of food, but if you look on this newt under a microscope, you can see that the skin is loaded with a biofilm of microbes, and it's actually microorganisms that are producing this toxin that is toxic to most other animals. But it's not toxic to the newt because the newt has a mutation in its own genome that kind of changes the shape of the sodium channels in the cell membranes. But again, so this is where the evolution and the two things come in. So it's not just microbes making it toxic, and it can just rub on these microbes and then it's toxic. It's because it evolved its own genome to then recruit these bacteria, or at least didn't get harmed by the bacteria, and then all of a sudden those bacteria actually gave it a trait that, to us, for many years, we would just assign that trait to the newt. It's a toxic or poisonous newt, but no, it's not. It's a holobiont that is poisonous. Okay, so hopefully now we have an idea about why holobionts are interesting to study. So I will just give a brief example from our own research in my group. So this is our model, very simplified. But the key is here we have a host organism, preferably inspired by your passion. And then this host organism has a bunch of microbes, and they're all over the place, but we very often look in the gut. But then what is really key is that we have these two arrows. We have both arrows. Because if all of the microbiome research you read about in the literature and the newspapers and the media, very oftentimes they're only focused on the right-hand arrow. What are the microbes doing to us? Are they making us sick, or how are they making us healthy? But what is really interesting, both from an evolutionary perspective, but also if you want to apply this in a more efficient manner in food production and health, it's that left-hand arrow that we're really, really interested in. So one example, we actually started out by making a growth trial of salmon in Norway. So we collaborate with some salmon farmers in Norway. I know, Maida, one day we'll all eat insects, but until then, we'll probably still eat a few fish on the way. So if we can make them more sustainable, why not? And accidentally, there was an outbreak of a bacterial disease called tuberculosis, which wasn't supposed to happen. But all of a sudden, we found our tanks with some healthy and some sick salmon. So we kind of renamed the project to Krabby Fish. We still took the samples. And then we kind of compared these healthy and sick salmon, because they were sampled at the same time. They were in the same tank. Same exposure to these pathogens, so we considered those that were healthy at the time of sampling were more resilient to this infection. So first we were looking at the correlation between the size of the fish, which was pretty much correlated with whether they were sick or if they were healthy. And then the abundance of a good bacteria that we found. So we know this bacteria is good for the salmon. And we found this very striking example or correlation that you can pretty much just look at the bacteria, and then you can say whether it's from a healthy or sick salmon, or the abundance of this bacteria. We also looked at a bacteria that we know is often pathogenic and not very good to the salmon, and here we saw the opposite, so a strong negative correlation between the bad bacteria and the size or the health status of the fish. So while these are just correlations that we observe, how we would like to interpret it initially is that it is what's going on is this left-hand side arrow, right? So in the healthy fish, The salmon has a completely well-functioning immune system, and it's able to control its microbes. So it can select for the good microbes and keep the bad ones out. But as soon as the salmon gets sick, it's stressed and it's immunity deprived, and it basically weakens the left-hand arrow, and that leaves space and opportunity for opportunistic pathogenic bacteria to emerge. So that's what we thought was happening. This is how we interpret in the lab and with my students, and it was pretty much just us. We wrote a paper, published it, a few people have read it, and we still think it's pretty cool. But what about real life? Why does it matter? So actually a few weeks ago, something cool happened. And it was so cool I didn't get any pictures of the event, but there was a fish veterinarian in Jutland, Denmark, who's an educated vet. He works for fish farms, and he had seen our paper for some reason, or heard about it. So he called me up. So it's like an unknown number on your phone that you rarely pick up. But for some reason, I picked it up that day because I had a feeling this was something good. And half an hour later, I was just blown away. So he told me, he goes out to these fish farms. They have these sick fish with the wounds all the time, the same infections. And they just don't like it. They have to kill those fish or try to treat them with antibiotics. And he didn't really know what's going on. And the big companies didn't really want to listen because it was too complicated to try and do anything about it except just start over. So what he explained to me is that all the healthy fish, so he's starting, when he get out there and there's a disease outbreak, he dissects the fish on the inside and the outside. And recently he started to also look in the gut. Normally he would not look in the gut if they have a skin wound. But then, so what he started to see is that actually when he puts the gut under a microscope, in the healthy fish, nothing's going on. It's just quiet, dead. Which fits with what we see in fish in general, so this is another thing, but in general there's just not a lot of microbes. That good microbes we see in the healthy fish actually lives inside the cell of the salmon, so you don't see it. But when he looks in the gut tissue of the sick salmon, he started to see what he called movable bacteria, so small critters that are just bouncing around, and he was pretty sure those were this Allivibrio kind of bacteria, which was exactly the bad bacteria that I showed up there. So it took a while, but basically from his empirical observations out there in the real world, dissecting sick and healthy fish, he observed exactly the same thing that we did in our molecular data. So it was really, I think we were both pretty stoked about confirming each ways about there's something to it. So hopefully we'll keep talking to him. He's a one-man company, but maybe we could slowly start to convince the rest of the people that we can use this research to understand these holobionts in a much more efficient and detailed manner. So on that note, I think I will end it here. Veronica.